Hello, this is Amanda Welch welcoming you to this Bite-Sized Bio web seminar, which today is sponsored by QIAGEN. QIAGEN is the leading global provider of sample-to-insight solutions to transform biological materials into valuable molecular insights. QIAGEN sample technologies isolate and process DNA, RNA, and proteins from blood, tissue, and other materials. Assay technologies make these biomolecules visible and ready for analysis. Bioinformatics software and knowledge bases interpret data to report relevant, actionable insights. Automation solutions tie these together in seamless and cost-effective molecular testing workflows. QIAGEN provides these workflows to more than 500,000 customers around the world in molecular diagnostics, human healthcare, applied testing, forensics, veterinary testing and food safety, pharma, pharmaceutical and biotechnology companies, and academia, life sciences research. Today's presentation is titled The Use of qPCR to Validate Epigenetic Enrichment of Pathogen DNA from Complex Samples and Human DNA from Stools, and is being presented by Dr. Alan Forsyth, Vice President of Infectious Disease at California Life Sciences Institute. Alan received a PhD in molecular biology from UC San Diego and has held founding and executive positions at Elytra Pharmaceuticals, Merck, Fleur, and Singlera Genomics. He has developed novel approaches to the isolation and analysis of rare cells, such as microfluidic chips, to separate circulating tumor cells from the blood of cancer patients. His current work is the result of a DHS contract to isolate pathogen genomes from complex samples. As always, we will have a question and answer session after the presentation, so please type any questions that you have into the questions box, which appears on the right-hand side of your screen, and I'll quote them to Alan at the end. And a recording of this webinar will be available at bit.ly/validate epigenetics enrichment webinar. That's bit.ly/validate epigenetic enrichment webinar, all one word, lowercase. So now over to you, Alan, for the presentation.
Well, thank you. And thanks everyone for logging in today to this webinar. As she mentioned, I'm going to talk to you about some of the uses of qPCR in our lab to validate epigenetic enrichment. The agenda that I want to go through is a little ambitious, but in our lab, we look at a lot of rare genetic events, and I want to sort of define those for this audience and talk about some of the difficulties that come up in handling those and some of the good reasons for enrichment. One of our favorite enrichment approaches is actually epigenetic, and I'll talk to you about what kind of epigenetic differences there are in microbial versus human genomes, and even within the human genome, that make this such a powerful tool. Probably one of our favorite enrichment tools are in fact restriction endonucleases, which is a relatively novel approach, and so I'll walk you through how we do that and some of the validation tools that we've used to show the power of this approach, notably qPCR, some NGS that we've done in order to look at how enrichment occurs in more complex and real-world samples. And then ultimately, the use of enrichment also provides concentration that really improves sensitivity. That's one of the main difficulties when working with rare genetic events. I'll also show how this can be applied not just to pathogens, which is part of the big title of this, but also to human DNA. That's the rare component in things like stool. So any talk like this is also important to at least bring up the issues of how contamination can creep into your experiments and how we avoid it. So with that, let me get started. So this particular chart is looking at a series of things that I consider rare events. A number of diseases fall into these categories, and their causative agent, the number of target organisms per cell, per mil that cause the disease state, and the background type of cells that you can expect. As one example that we won't get into a lot today, UTIs, or urinary tract infections, are predominantly caused by E. coli. And as soon as they reach a cell number of about 1,000 per mil, this can cause problems. This is technically an infection. There's almost always some trace levels of human cells, but I think this audience appreciates that even one human cell has 1,000 times the amount of DNA mass as a bacterial cell. So just one cell is essentially an equal mixture of 1,000 bacteria versus one. And as you start to get to tens or hundreds of human cells, the pathogen becomes the rare genetic event. This kind of problem only gets worse in most of the disease states that are listed here. These different intracellular pathogens like Lyme disease, other spirochetes, tuberculosis, rarely have much more than 10 copies in a mil of a clinical sample. But the backgrounds tend to be 10 to the fourth human cells. So really this ends up being more like a 1 in a million to 1 in 10 to the seventh kind of problem. And one of the more challenging instances that we look at in our lab is detection of the causative agents of sepsis. And in those cases, we're really looking at 1 in a billion kind of a problem. As I mentioned towards the end of the talk, I'll take a look at cases of human diseases, notably colorectal cancer, but really any liquid biopsy sample rarely has more than, say, 5 circulating tumor cells. And if it's blood, your background is going to be 10 to the seventh normal cells. And of course, the rare event here is somewhat different, right? You're really looking at the ratio of a mutation to the wild type. And whether it's circulating tumor cells or circulating nucleic acids, common tumors that people are looking for rarely hit the 1% level and can be even much rarer than that. So epigenetics is really the modification of the standard 4 bases of DNA. And this table looks at some of the common epigenetic modifications across the different kingdoms, from bacteria, protists, animals, plant, fungi. Again, we're all familiar with the standard 4 bases, A, G, C, and T, and most people are very familiar with 5-methylcytosine. So the methylation occurs at this 5 position. But what's not as commonly appreciated is how many of these bases are stably maintained in the host, passed on to progeny, and especially in bacteria, become very frequent, maybe as often as 1 in every couple hundred bases in the case of N6-methyladenine. And so that provides what might almost be called a 5th, 6th, or 7th base of DNA. And the reason I consider this to be important is that almost all rare event biology is about signal and noise. And what you're looking at here are signals that are often not present in one kingdom or one biology state versus another. And so if you have a good tool for fishing out that signal and you have essentially no noise in your background, you've got a really good tool for doing an enrichment. So what you can notice here is that in addition to there being several other bases or modified bases that are present in the bacterial kingdoms, N6-methyladenine, N4-methylcytosine, they have unique roles in the bacterial kingdom: replication, DNA repair, and restriction modification. And we've taken advantage of some restriction enzymes to do enrichment. So I want to take a moment just to talk to you about the restriction modification system. And there's nothing new about the restriction modification system. In fact, these are enzymes that probably evolved near the beginning of life, some couple billion years ago. And bacteria quickly began to do something to their DNA, and that is to methylate certain bases. And in doing that, they sort of created this epigenetic mark that allowed them to distinguish self from non-self. And in making a methyltransferase to place that epigenetic mark, they would make a cognate restriction enzyme that would digest any DNA identified that did not have that cognate mark. So what you're looking at in this box is essentially a billion-year experiment to create a molecule, an enzyme, that has extremely high discrimination for target versus non-target. In other words, it makes for the perfect tool for collecting an epigenetic strand of DNA. The other thing about restriction enzymes that makes them particularly useful for our uses is how rapidly they work. So, as you can imagine, they had to evolve to sort of rapidly scan all the chromosome and rapidly identify an invading phage, otherwise the cell dies. That's why I think this is such a great evolutionary tool. So endonucleases are also very quick to load onto DNA non-specifically, track along the DNA by facilitated diffusion, and when they recognize their site, they kink it, exposing the phosphodiester bond and allowing catalysis to digest and essentially break the DNA strand. So what this group needs to know is that catalysis is actually very easy to inactivate, while the binding, diffusion, recognition are all easily maintained. And the enzymes are actually also easily manipulated. For instance, you can biotinylate them and add them to streptavidin beads, magnetic beads. So that's exactly what we've done in our lab. So how do you use restriction endonucleases as enrichment tools? Well, as I just said, the wild type DPN1, depicted by this sort of green dot, would load and scan DNA until it sees its recognition site, which in this case is a GATC motif that's methylated at the adenine at the N6 position. Upon seeing that site, recognizing that site, kinks it, cleaves it. So what we've done, as I mentioned, is just adhere them to magnetic beads. And we can inactivate the catalytic activity either through mutations, modifications of buffer to remove cofactors or poison cofactors. So these features allow us now to have a molecule, a magnetic bead, with multiple endonucleases attached that has high discriminatory capabilities. And so the cartoon suggests what's going to happen. These magnetic beads will load onto the DNA, scan until they find the recognition site, bind specifically to the methylated motif, but they don't go through catalysis. And so now one can use a magnet to fish out the DNA that's bound to these magnets, wash off presumably DNA that you don't want, and get an enriched product. So how does this work? The first time we tried this, we did something, I don't want to say crude, but we used an agarose gel for analysis. And we used DNA products, as you can see on this cartoon. One was a 650 base pair product, and the other was a 477 base pair subset of it. And all we did was one of them had the GATC site methylated, the bacterial fragment, and the other did not. So we could mix the two and differentiate them on a gel based on their size. So you mix them, we add in our DPN1 coated beads. That's all in a tube. What washes off should be the non-bound, unmethylated 651 base pair fragment. What we collect by magnetics should be the target fragment, and when we elute, we expect to see the 477 base pair fragment. So in the gel, you can see the initial input, the two fragments here, a 651 and a 477. As we titrated in magnetic beads, we took the soup and looked what was in it, and you can see that 651 fragment that is still in the soup. And when we elute off the beads, we got a nice clean fragment back. So we patted ourselves in the back, felt like we'd had a tool that we can use, but realized we wanted to do something a lot more quantitative. And so that's where qPCR is such a great tool. And so I'll give you a rundown of a particular assay that we used for this, and how did we generated our standard curve. So here you can see that we had a set of primers specific to the 16S of E. coli, forward reverse primers, and a good probe. We used a very standard PCR reaction using QIAGEN master mix, our probe mix, water, and in this case, just E. coli DNA in the titration. And we ran the titration through a PCR cycle. And what you can see is we titrated down the amount of E. coli DNA as we got a nice standard curve with a good R² value. So we knew that we had a good sensitive method to detect the presence of E. coli, and you can see our zero control down here at the bottom going out some 30+ cycles. So how do we set up an assay or a test then to look at that? In this particular test, we've included a 6-point titration series, and each one of these data points is a mixture that includes 10 micrograms of human DNA and some amount of E. coli DNA in decreasing amounts in each of the 6 tubes, down to 10 femtograms. So when we make this mixture, we use our E. coli primer probe set. We also have a human E. coli probe set. And we measured the inputs to generate these two curves. You can see the constant amount of 10 micrograms of human DNA in each of the tubes and the decreasing amount of E. coli DNA in each of the tubes. And notice something, that our standard, essentially it's still a standard curve, right? In the presence of 10 micrograms, we begin to lose a certain amount of our sensitivity. So this is not an uncommon feature. Once you start to have a DNA molecule that's present at 10 to the 7th, 10 to the 8th, 10 to the 9th levels lower than the background, qPCR does get more complicated. But you can see how linear it is across 5, 6 orders of magnitude. Now, the other thing that we did was we took each of these tubes, we added our DPN1 bead mixture, mixed all of it, incubated, put it on a magnet, washed off the unbound fraction, and then analyzed the bound fraction. And here's what we observed. While this blue line indicated the amount of human that was input, this was the amount of human DNA that we recovered. In other words, most of it washed off. And you can see that we lost over 2 orders of magnitude of the input human DNA. On the flip side, the E. coli DNA, shown here by the dotted line, was recovered at over 80%, all the way down into the femtogram range. So we know from these kinds of experiments the following kind of conclusions: that at least with our primers and probes, the 16S marker of E. coli is incredibly efficiently recovered by this kind of enrichment technology, from a nanogram down to femtogram levels in a fairly high background. This enrichment works across at least 5 orders of magnitude, although we recognize that our absolute sensitivity at these lowest levels is a little bit hampered in the presence of a very high background. We can also say that the human DYZ marker is efficiently excluded by about 99.6% in most of these tests. But we're only looking at these 2 markers. So we have to look at other tools to examine whole genomes and look at more complex mixtures. And that's in the next slide. So using next generation sequencing, we decided to make a more complex genomic mix. And we used 12 organisms that are listed down here, and we put them in pairs. So for instance, Homo sapiens and Oryza sativa, which is rice, were mixed in at sort of the top level. Then we did a 10-fold dilution and put in the pair of Aspergillus and E. coli, and another 10-fold dilution. So all of these are decreasing in their concentration by 10-fold decrements. And that's a little easier to see when we've actually sequenced it, how it came out. So this graph on the horizontal axis is showing you the input number of sequencing reads. So for instance, for human and rice, we had put in something like 5 million reads into the NGS experiment. We were angling for 10, we got a slightly lower amount. Or I'm sorry, 5 million reads. But then you can also see that in the enriched or bound fraction, we only recovered back 500,000 reads or hundreds of thousands of reads. So it was greatly depleted in the enriched fraction compared to the input fraction. And in fact, this dotted line shows you what the input fraction should look like. As you go down this graph, you can see how the other pairs of organisms fared. Aspergillus and E. coli, for instance, Aspergillus went in at 100,000 reads, came out at about the same level, whereas E. coli, we suddenly had several million reads. And what we observed, and we kind of expected something like this, but it came out much more striking than we expected. That is that there's essentially 3 types of methylomes in this experiment. All of these organisms that are well above the line are all organisms that contain GATC. They include the DAM methylase. Their genomes are methylated at GATC sites that occur maybe every 250 bases throughout the genome. These microbes along the line do not have methylated GATCs, and so they don't show the same level of enrichment. What's particularly striking, though, is that human and rice have higher eukaryotic methylation patterns, which includes not only the absence of GATC methylation, but the presence of extensive methylation. And so there's a dislike of the enzyme for binding to these, and you see a decrease in their recovery. So this particular experiment showed us a lot of different things, in particular, that this process is not only sensitive to very low dilutions. Notice the levels that things like Vibrio cholera are spiked into, 5 orders of magnitude lower, but also that it's effective across a broad range of genomes. And even these other microbes are enriched an order of magnitude compared to plants. What this graph doesn't tell you a lot about is how well the coverage occurs across each of these genomes. And I'm just going to look at E. coli as one example on the next slide. So next generation sequencing is nice in that we're looking at essentially all markers of the genome. And you can see this is the input. This is the number of reads on the vertical axis and the number of reads achieved at each position starting at an arbitrary 0 out to 4.6 megabases of the E. coli genome. And what you see is that the input looks pretty even. There's a blow up here on a different scale. And that's compared to post-enrichment in this lower panel. And you can see now that these two are on the same scale that it dramatically increased, to 250, 300,000 reads across much of the genome. The other thing that you can notice is that the pattern that was observed in the input is maintained in the output. And this was actually pretty cool to somebody like me who does a lot of bacterial replication experiments. The E. coli genome is circular, and so you see more of the origin as it's replicating than you do of the terminus. And those physical features of the genomic representation that were there on the input are even present post-enrichment. So enrichment allows us to get much better coverage. It allows us to analyze things like pathogenicity markers. We can actually see physical features of the genome. And we kind of walked away from our sort of synthetic mixture of genomes experiments and said, it works. Now let's try it in the real world. And we've done a lot of these kinds of samples. We've published a number of them. I think Our publications are readily available. But I'll show you a couple examples. This first one on the far left is saliva, and we're looking at a pooled human sample that has gone through enrichment. So the input is not enriched, and again, this is out to 1 million reads or so, normalized actually to 10 million reads. And on the vertical axis is the enriched bound fraction, and you see immediately the same kind of thing. There's a number of genomes that are dramatically enriched. We've highlighted a few that are known DAM organisms, that is, that they have GATC methylation. There are several organisms that were not even detectable in the input that suddenly become highly enriched. And there are a few that are somewhat depleted. I think it was brought up at the beginning of this talk that this work was funded by Homeland Security. They're interested in looking for pathogens in various kinds of complex samples. So in support of their work, we not only looked at human clinical samples, but we also looked at things like creek water samples. This was a polluted lake, and they were a river, and they wanted to see what kinds of organisms were present. And in the absence of enrichment, we were getting fewer than, say, 10 reads in the input in this kind of a sample. And post-enrichment, there's hundreds of reads of a pathogen like Legionella that were present in this creek. So the other thing that I wanted to share with you about enrichment is some of the benefits that include concentration. And this is particularly important because not all samples come in a 10 microliter volume. And blood is a great example of something like that. This is a 3 ml tube of blood, I believe we were working with. And one of the first things that you might do is use a DNA kit to isolate DNA. And now you might elute in something like 100, 200 microliters. But anyone who's worked with blood DNA knows that it's difficult to put a lot of blood DNA into a qPCR reaction. You tend to get inhibition. And so you can really only put a couple microliters in, and then you start to decrease your signal. So in most cases, we might do 2 microliters of blood DNA into a fairly large PCR reaction, something like 50 microliters. And when we did that, and I think I forgot to mention that this tube was spiked with about 200 E. coli cells. So 3 mL of blood, 200 E. coli cells, isolate the DNA, take 2 microliters or 1/50th of that, and now we can run a PCR reaction just straight away, no enrichment. And you would expect something like 8 cells worth in there. And indeed, we get just under 10 cells. We get a signal that looks to be the equivalent of about 10 cells. If we do enrichment, we see the same kind of thing. So, that's good. Enrichment works. But one thing that you can't do with the straight blood DNA is put all of it into a reaction. 100 microliters of blood DNA is a very large qPCR, very expensive qPCR reaction at best. But what we can do is when we do our enrichment, we can pull off the soup and resuspend the beads directly in the qPCR mixture. And then when we do that, we can just do it in a standard reaction as we did here. And suddenly we get a much stronger signal. I note that we did not get back the full 200 cell signal that we would have hoped for, but we get back a very strong signal. And especially when you're looking for things that may have only been present at, say, 50 or 10 cells in this tube, you would have completely lost it if you had not been able to stuff the entire blood volume of DNA into the reaction. So some observations, the direct resuspension of an entire volume of DNA into an enrichment reaction allows us to do pretty extreme concentrations. In this particular case, we've put in as much as 500 microliters and resuspended it in 10. In this next example that I'll show you, I think it was a mill reaction. One nice thing is that we believe we see reductions in blood DNA inhibition. That's a set of data that I'm not going to show you today, but it's sort of apparent. And certainly, we're removing hundreds of micrograms of human DNA. And as you saw in earlier slides, when you get rid of that human DNA background, you improve your sensitivity at low levels. So the other thing that this becomes important for is really whenever you're working with clinical samples, concentration can be incredibly useful. And that's always an issue when you want to look at the human methylome. So why does one want to enrich human DNA, not just pathogen DNA? Human DNA is sometimes the minor component. It is sometimes present at low quantities in samples like stool. Tumor DNA is also typically present at low concentrations, often well below 1% in liquid biopsy samples. And targeted enrichment of the methylome is a great way to improve your diagnosis of different disease states and enhance discovery of different methylation-based diseases. So for the following experiments, we're not using DPN1, but we're using a type 4 enzyme, MCRA, that recognizes the following motif. A CG motif where methylation occurs at the C, 5-methyl-C. So standard human methylation pattern. And there's a slight preference for pyrimidine, cytosine, and tyrosine on the front end and purines on the back end. And I'll show you some examples that we looked at to examine liquid biopsy's efficiency in the presence of stool. So the lab instantly gets upset when you say you're going to work with stool samples. Why would anyone want to examine stool? And there's actually a pretty simple answer. People really don't like colonoscopies. So, so there's a motivation to identify mechanisms that we can test human health without being as invasive as a colonoscopy. But this still has a lot of challenges, right? It may be relatively simple for a patient to collect the sample, but it needs to be stabilized, transported, and then in the lab, you're going to do homogenization, DNA isolation, PCR, and maybe some kind of sequencing or detection. But there's a lot of challenges. Once you get those samples, homogenization strategies are challenging with something that's relatively non-homogeneous. Isolating DNA is difficult, and we find that we often get very low quantities, very low concentrations of human DNA, and we have to dilute our samples because of the presence of stool inhibitors. The inhibitors are many different kinds of things. There's carbohydrates and other kinds of things there, but there's a huge excess of bacterial DNA. So when I talked about rare genetic events, human DNA is the rare component in stool. And when you try and do a standard curve, in this case, we like to use ALU. ALU sequences are scattered frequently throughout the human genome, so you've got a nice strong signal. And it gives us a very good standard curve. But when we do that in the presence of stool, all our curves compact down. And we do dilutions and different things, as I mentioned, but there are a lot of challenges in actually getting an accurate determination. If we do NGS, and this particular sample is a great example of humans working together to do science. This individual donated a sample after eating only sequenced organisms for a week. This is an experiment that was done some time ago when we didn't really know what to expect in sequencing a stool sample. But what we found out was that the human fraction was an incredibly small component, let alone trying to detect anything that you might have eaten. It's dominated by bacteria. So we envision a slightly alternative workflow to what I suggested before. We might collect a couple grams of sample to homogenize, but well under that, maybe a couple 100 milligrams will go into DNA isolation. We'll take 100 microliters of isolated DNA and dilute it to 1 mil in an enrichment. And this gives us the opportunity to dilute out the inhibitors, which are in the soup, whereas the DNA is going to get bound to our beads, wash everything else away, and resuspend those beads in 50 microliters of PCR for detection, or move on to a library prep. So what do we get from this? As I mentioned, we might put in something like 2 grams of stool. We'll get something back like, say, 86 nanograms of human DNA. It doesn't all go in to the enrichment, but if we put in something like 11 nanograms, we get back 82% of it. So we have a very good yield using this process. The amount of bacterial DNA will be in the microgram levels. We'll reject 96% of that. And so it gives us a pretty good ratio. You know, you come in at ratios that are well under 1% human DNA, and you come out at ratios that are more like 35%. We don't have published NGS data yet, but we do have some good conclusions. Notably, that it is possible to segregate human DNA from a large excess of bacterial DNA. This can be done in the presence of a very complex and difficult to work with matrix in this case. And we can concentrate a very large sample for direct use in PCR. And we remove a lot of the PCR inhibitors. And we assume that this is because an enzyme is not at all like silica. And so there's a lot of things that stick to silica because of its charge state that just doesn't happen with a restriction enzyme. And as I said, kind of the bottom line is we end up with about a 35% human DNA concentration, way better than 1%. And so that puts us in a reasonable position to do CRC mutation profiling. So I mentioned I would at least touch on the topic of contamination, and let me wrap up some of the data with these comments. We have seen tubes and tips come in with contamination. We have seen magnetic beads come into our lab and they're contaminated. We haven't seen anything with primers and probes, but we have contaminated those ourselves. So you can imagine some of the suggestions I'll be giving this audience. Enzyme preparations have typically shown up clean. Buffers have gotten contaminated. I mean, lab personnel often introduce it, and your environment often introduces it. So really, you have to be suspicious of everything. And the solutions, of course, are to process and to have a process that you're going to test essentially all vendor components in each new lots. When things come in, we test the lot, we'll break it into aliquots, test the aliquots, freeze them down. And now we know we've got, you know, good sterile components that we can go back in and pull out when we're ready for them. You should have a standardized workflow where you separate amplification. Sample handling is a completely different area in our laboratories. Likewise, NGS is separated off. Wherever you can, you design primers and probes to minimize amplification from humans and commensals. I mean, that's part of the power of qPCR. And we do a lot of training, and personnel are tested on assays that are prone to contamination. You know, sensitive human assays like DYZ and ALU are great, not first test for your new personnel, but sort of a last test before you move them on to more sensitive assays. And of course, all of these things should have no template controls with them. So probably nothing new there, but it's probably helpful to hear that other people have had things come into their lab, and these are the ways that you catch it and prevent it from ruining an experiment. So conclusions for the use of endonucleases for enrichment. First, I should point out that the eukaryotic kingdom, really the signals are in the DNA world are AGCT and methylated C. You have other signals in the bacterial kingdom. So this gives you opportunities to choose your signal and noise, especially because there are over 300 restriction endonucleases out there. And so that gives you a lot of options for what you want to bind and not bind using these kinds of approaches. I think I only showed you 2 restriction enzymes in today's work. I think we've published on maybe 5 or 6. This technique we've shown is sensitive to small fragments. The smallest one I showed you today might have been that 477 base pair fragment in kind of the agarose gel. I think we've done gel shifts on fragments of about 20 base pairs. And I showed you several cases where we're sensitive to femtogram quantities, or essentially a single cell's worth. We actually think we probably are have about a 70% chance of binding each fragment in a mixture. We can demonstrate genome-wide sensitivity. There aren't a lot of hot spots or cold spots. And we can segregate or enrich a target DNA from a large excess of non-target DNA. This process can be done where we can concentrate an entire sample, hundreds of microliters up to a mil, for direct use in a PCR reaction. And we see that at least some PCR inhibitors are removed during this process or reduced in a way that is not like silica. So with that, I'll say thank you and return to our sponsor. But I will also give thanks to our funder in this case, who is Homeland Security for this work, and my many fellow co-workers who will appear in my publications. So thank you to this audience.
Thanks, Alan. That was an excellent presentation. We do have a few questions from the audience, but if anyone else has a question, please feel free to post it in the questions box that appears on the right of your screen. So the first question I have is, so how is detection done today for disease states like sepsis, where the genome target is present at about 1 in 10 billion?
Interesting question. Presently, in clinics and the like, they are not using a lot of molecular biology for this. This problem is still done primarily through microbiology. So samples of about 3 mils are plated on agarose plates, and essentially you're amplifying the signal through growth. And so you'll hear people complain that they can't get information about what the infectious agent is for 3 days to a week, however long it takes to grow up the strain and then type it. So really the answer to your question is there's not good tools. There aren't good rapid tools when you start to have a 1 in a billion problem. And that's exactly the problem we're trying to solve.
That makes sense, yeah, because that's such a small ratio that kind of blows my mind trying to figure that out.
Yeah, it's also not easy to even do experiments like that, right?
Right. Yeah.
My team does a great job with some of the titrations. They look clean and nice, but it's not always as easy as it might look.
Okay. And our next question is, this is a very intriguing enrichment method, but did the biases that you observe cloud your ability to analyze a complex mixture?
Well, that's an interesting question because I think by its nature, enrichment is a bias. You're certainly you're picking winners and losers.
Right.
If you understand what the bias is and it works in your favor, then it's a good method. Actually, could you repeat that question again?
So this is an intriguing enrichment method, but do the biases you observe cloud your ability to analyze a complex mixture?
Okay. So I think the answer is no. When you know what the biases are, you make them work in your favor. If you don't know what a bias is, then you'll have something hidden from you, and it could cloud your judgment. So I guess I do get questions about biases from time to time, especially from the microbiome community, because they don't want to disturb the ratios of the bacteria that are present in a sample. And this will certainly enhance the representation of some bacteria over others. But we want to do that. So we're trying to pick the needle out of a haystack, and we're choosing enrichment tools that enhance our ability to look at certain kinds of pathogens. And that does mean that there's a bias against the others. So interesting question. And I think it always depends on what you want to do as to whether you call it a unfortunate bias or something that's actually working in your favor.
It sounds like kind of these bias or this bias depends on what your desired outcome, like what you're wanting to study.
Right, right. So there is, I guess, there is a risk that you could miss something that you didn't know was there. But if you took advantage of some of the samples that we put up on the board, like creek water, I mean, that biome is just rich with thousands, if not millions, of organisms present, and it's pretty clear that we're detecting a number of things that you couldn't do without this technology. Are there other things there that are being lost? It's a hard question to answer, because if we just use standard techniques, we don't see them. So there's a bias, yes, but you never know what you can't see.
Right. You can't know what you don't know. And then I have a question about, so which do you use more, qPCR or NGS for analysis, and why?
I think we use qPCR more. It's faster. You get your answer, a couple hours later. It's relatively inexpensive. And if we know what we're looking for, or we have a good surrogate, it pretty much tells us if something worked or didn't work. But when you want to do more than analyze a few markers, then we get into NGS. And that's often the case, I don't know, when it's an unknown agent, or we want to characterize something fairly thoroughly. If we want to do more than just look at predetermined markers. So discovery work is almost always a little more NGS based.
Okay. Yeah, that makes sense, because you'll be able to get a wider target or a wider range of results.
Right, and just a greater depth of information. We were just talking about what you know versus what you don't know. If in our lab we're looking at a pathogen, and we might set up qPCR primers to see if methicillin resistance is present, and maybe it's not. But if we didn't use primers to test for carbanicillin resistance, you won't know if it's there or not there. But if you do sequencing, you'll read out everything that's present. So there's clearly advantages sometimes to NGS. The biggest issue is time and cost.
Yeah, that makes sense. And then our last, so we do have, okay, sorry, we have a question that just came in. So Igor asks if you have any recommendations for the blood volume for DNA extraction in the case for a pathogen detection and quantification through qPCR.
The problem with blood samples, and really any biological or human sample, is they're so varied. So depending on the pathogen you're looking for, it may or may not be present in a 1, 2, or 3 mil volume. At the early stages of a bloodborne infection, I'm not convinced that even 10 mils will have something at the very early stages of an infection. So that's kind of a tough question to answer. The standard of care is to culture 3 mils. Okay. So I think that's the safe answer. The second part of his question might be a little more technical. How much blood can you put into a DNA isolation, and how much can you get into a qPCR reaction? We've been working with kits that allow us to isolate DNA from 10 mils, and we're getting it all into a single qPCR reaction. But we are doing a lot of in-house tricks to make that work.
Okay. I was kind of surprised that's a very large quantity of starting material.
Yes, it's not trivial.
Okay. And then we have a question about how are you, in particular, how are you applying your technology?
Well, that last question was actually a lead-in.
Okay.
We are a group that doesn't mind taking on a big challenge. So we have been looking at trying to be able to do detections of bloodstream infections early in the disease state. I'm also working with a collaborator to detect treponemes and to do sequencing of treponemes. These are spirochetes that cause things like syphilis and related diseases. To do whole genome sequencing. Treponemes are not culturable. So non-culturable organisms that are present in 1 in a million type quantities are pretty hard to sequence. And we've been working with the collaborators to sort of do this from a wide variety of sample types. So you'll see a publication on that probably during the course of this year. And I hope to be able to publish more of our work on bloodstream infections in the future.
Okay, and we have a question from Ruby, and they ask, how do you identify the different types of contamination versus aggregation?
I might have to have her clarify that, but I think she's asking, how do you know where the contamination comes from? The second part, asking about aggregation, I'm not sure what she means by that. So I'll tackle the first part. There's sort of two clues. One is, what is the contaminant? And that can sometimes give you a clue. Whenever I see human commensals, P. acne, and things that are on our skin, I don't blame the lab staff, but I suspect that it's personnel that inadvertently contaminated something. When we see things that are a bit more esoteric, different pseudomonid species, things like that, those are more commonly coming from buffers and the like. The way we figure out what it is controls, and then we might have to go back and test 2 or 3 buffers, something like that, in order to identify the specific component that we're going to toss out. And sometimes we just toss out 2 or 3 components and bring out all new vials for things.
That makes sense, because sometimes it's just better to just do that.
Yeah, sometimes it's starting fresh. And like I said, I don't know how clear I was around this. Essentially, we validate every component up front. And that way, when we do see a contaminant, we know it's not sort of rampant in all of our materials, and it's much easier to track down.
Okay, and then we have a question from Dickie. They ask, is there any gene related to sepsis? Is there any gene related to sepsis that can be used for detection? I believe is what he's saying.
Any gene related to sepsis that can be used for detection. That might actually have 2 components to it. One is, if it's a bloodstream infection, the causative agent can be one of a large number of things. I would imagine that there's about 25, 30 organisms that make up about 95% of infections. So that's a pretty big range of markers to look for.
Yeah.
There are markers of sepsis, not necessarily the infection, but the actual state of the septic response of the patient. So there are a number of those markers out there, and you can probably just Google and take a look at some of them. But when it comes to the actual identification of the causative agent, using 16S and 18S and these sort of universal markers for microbes is about as simplistic as you can get, although it won't necessarily give you a specific ID, it'll give you a generic ID.
Okay. That does make sense. Okay, and I believe that is our last question for our Q&A session today. So to close out, QIAGEN has designed a checklist for the efficient pathogen detection, as well as 2 detection kits, all designed to help you with your research. The QIAGEN QuantiNova pathogen kits contain the complete list of safety features, such as in-process controls and 2-phase hot start to assist researchers in overcoming their challenges during pathogen detection. While the QuantiNova pathogen kits are dedicated for pathogen detection, the QuantiNova qPCR kits support any gene expression experiments. Both kits are automation ready if needed. To learn more, please visit the QIAGEN PCR gene expression page. And you can look at that under the resources in the knowledge area for RNA Universe /gene-expression/PCR. And the link for this is being sent out to everybody right now. Well, and that brings us to the end of our seminar. So thank you again, Alan, for a very illuminating presentation and a great discussion. And thanks also to our sponsor, QIAGEN. And finally, thanks to you, the audience, for taking the time to attend and listen in. If you've enjoyed the seminar and would like to view the video recording of the session, please visit the webinar page on bitesizebio.com. It should be available within the next 24 hours. There you can see the other webinars we have lined up for you in Bitesize Bio. So until next time, good luck in your research, and goodbye from all of us at QIAGEN and Bitesize Bio.